Okay, so welcome everyone. Let's get started. So today, I think we have a treat. It's my really great pleasure to welcome today's speaker, Dr. Feng Shao from NIBS Beijing. And I think most of us actually know Feng, or at least his work very well. So currently, Feng is the Deputy Director of National Institute of Biological Sciences, which is NIBS. So I think I just to give a brief introduction. So Feng actually finishes undergraduate study in Beijing University, and then further pursue his PhD in the University of Michigan. From 2003, he did a very brief postdoc in UCSD and following up with another postdoc in Harvard Medical School, which also totally approximately 2 years. So after that, Feng moved to NIBS, started his career and with his own independent laboratory. So from 2005 until now, so Feng stayed in NIBS and then start from assistant investigator to full investigator, and now it's the deputy director of the NIBS. So I'm not going to go to dive in his publications because it's just too much. I think his total publication is more citation, over 20,000. I think it's really remarkable. I think Feng's lab is mainly interested in studying molecular mechanism of bacterial infection and host innate immunity defense. At the same time, he also studied how the host uses its innate immune system to react with bacterial infection, particularly the inflammasome pathway in macrophages. His lab also combined different approaches, including biomedical reconstitution, cell biology, and mouse genetics to identify new components in pathogen-induced inflammasome activation, and then to further review the underlying machinery. It's really remarkable how many recognitions, how many titles. I think I'm just going to give a few of Feng's achievement. He's a member of German National Academy of Science, also a fellow of Chinese Academy of Medical Sciences, also a member of Chinese Academy of Sciences. He really has a lot of honors and awards. I think mainly I just mentioned that he's the recipient of the second National Award of Excellence in Innovation in 2020. He also got Future Science Prize in 2019. And then he's also the awards recipient of Heliang Heli Foundation Awards. I think one thing I particularly would like to emphasize is his lab is really culture a lot of great students and postdoc fellows who's now actually start their independent research in different area and different institutions. I think nowadays it's particularly appreciated because I think we all need a mentor like Feng give a great guidance and then also share the great sciences. So without further ado, I think I'm going to give Feng's platform, and I think he's going to talk about pyroptosis in antibacterial and anti-tumor immunity. So welcome. Thank you for doing this, Feng.
Oh, thanks, Chuan. And it's really a great pleasure to have the opportunity to share our work with you, since we cannot see each other or meet each other in person. So if you have, among the audience, if you have any questions about my presentation later on, you can just drop me an email, so I'll respond. So today I would just focus on my talk on pyroptotic cell death. And this cell death was originally observed in bacterial infection. And then the concept of this cell death has evolved quite a bit in the past few years. And now we have some new data suggesting that this cell death may also induce antitumor immunity. So and in this talk, I'll divide my talk into 3 parts. And the first about 10 minutes, I will give you some background and introduction about innate immunity, inflammation, and pyroptosis, and the discovery of gastromin D. And then in the second part, I'll discuss our recent work on how pyroptosis mediated innate immune defense against the bacterial infection, and how some bacterial pathogens have hijacked this pyroptotic death pathway to evade from host immunity. And lastly, I'll talk about the role of pyroptotic cell death in cancer and cancer immunity. In that part, I'll mainly talk about gastromin E and gastromin B. So in innate immunity, we all know this Toll receptor pathway, and you probably don't remember that the Toll pathways, Toll receptors are membrane-bound proteins, and particularly for those Toll receptors that recognize bacterial products, they're only a plasma membrane on the cell surface. So in that regard, the Toll receptor can deal with extracellular bacterial pathogens. However, we have another group of bacterial pathogens such as tuberculosis or Salmonella, Shigella, and those bacterial pathogens, they were invaded into the cytosol space. So in that context, the Toll receptor will not be able to recognize intracellular bacterial infection. So starting from about 2007, the central question in my lab is how does the innate immune system deal with or recognize bacterial infection and initiate a innate defense response, particularly in the cytosol space. So here I'm going to show you a movie recording a very simple experiment. In this experiment, if we infect macrophages with intracellular bacteria like Shigella fraxineri or Salmonella typhimurium, and at the same time we add some PI, propylene iodide, into the cell culture medium to report membrane disruption or report lytic cell death. So as you can see, the macrophages do not uptake a PI. However, after being infected by those bacteria, the macrophages, they uptake propylene iodide, suggesting that those macrophages undergo explosive lytic death. And this is the pyroptosis that I'm going to discuss today. So basically, macrophages, once infected by intracellular bacteria, invasive bacteria, their plasma membrane integrity was disrupted. And then the DNA dye went into the nucleus, and the nucleus is already overstand, and the whole cells turn into red. So the cells are dying. And many other bacterial infections will cause more or less a similar kind of pyroptotic cell death. So not only bacterial infection, even some endogenous DAMPs, we call it danger-associated molecular patterns like ATP. If you treat macrophage with extracellular ATP, the macrophage will also die out of pyroptosis. And this phenomenon was early discovered in 1986 and later on in 1992. But for a very long time, and this cell death was misregarded as a bacterial valence mechanism. However, in fact, it's actually the opposite. It is not the bacteria that want to kill us. It is our cells that sense the bacterial infection and then commit the cell death, to induce our immune response. So basically, the cell death is not a bacterial valence. It's not a weapon of bacteria, but it's a host immune defense response. And in I think it's 1996, and Arturo Ziklinski's lab discovered that this cell death requires a caspase called caspase-1. And that brings another misunderstanding about this form of cell death. So caspase-1 actually was first discovered as an enzyme to process interleukin-1 beta in 1992 in this two very beautiful Science and Nature paper using biochemical fractionation approach to identify the proteolytic activity that is responsible for cleaving the precursor of interleukin-1 beta. That was caspase-1. So caspase-1 was first discovered in the context of inflammation and immunology. However, 1 year later, Bob Harvey's lab at MIT cloned a gene in C. elegans called SES3. So this SES3 is required for apoptotic cell death during the worm development. And at that time, there is only one homolog in mammals that is homologous to SES3, which is caspase-1. So in the 90s, with the progression of molecular biology, then numerous caspase were then cloned subsequently. But however, because of the discovery of SES3, which is equivalent to caspase-9, it was a critical role in apoptotic death, and then the entire caspase family, for some reason, was mislinked to apoptosis in the 90s. So now we know that apoptosis is probably more involved in in the cell death or in apoptosis during animal development. For example, like this tadpole development into frogs, and the tail is eliminated by apoptosis. So, so apoptosis clearly plays a key role in removal of cells and tissues during normal animal development. So if this cell pyroptosis, I just show you that as a response, as a host immune response to bacterial infection, then caspase-1 needs to be activated by an innate immune sensor pathway, which is called inflammasome pathway. And I believe many of you have touched on this. And the canonical inflammasome pathway is mediated by a member of the null-like receptor family, and among like NRP3, which has been heavily studied in the past 10 years. So the null-like receptors, they basically serve as a receptor in the cytosol to recognize bacterial products or some danger signals, and then recruit adaptive protein ASC, and then further recruit the pro-caspase-1. Pro-caspase-1 become activated into this tetramer active form. So we have characterized several inflammasome pathways in the past 10 years. For example, we identified the NIBS, which is a subfamily of null-like receptor that serve as a bona fide, in a biochemical sense, are receptors for bacterial flagellum and components of the type 3 secretion apparatus in gram-negative bacteria. So, and then active caspase-1 will trigger cell death in addition to process interleukin-1 beta, and as well as interleukin-18. So this cytokine response also suggests that the pyroptosis is a form of a pro-inflammatory cell death. Indeed, among all the cell deaths we now know, pyroptosis is probably the most inflammatory, pro-inflammatory form of programmed cell death. In addition to caspase-1, there is another caspase called caspase-11 in mice. In human, is caspase-4 and 5. And Vishal Dixit's lab in my lab, in the last few years, we characterized that caspase-11, 4, and 5 are indeed intracellular receptors for cytosolic lipopolysaccharide from gram-negative bacteria. So the lipid A moiety of LPS will be recognized by the CAR domain, of caspase-11, and that will induce oligomerization of caspase-11, and eventually caspase-11 will also induce cell death. And there are two important differences of the caspase-11 pathway from the caspase-1 pathway. First, the caspase-11 pathway does not process the interleukin. It only triggers pyroptotic cell death. And secondly, the caspase-1 pathway is mainly present in macrophage or monocytic cells, but the caspase-11 or caspase-4 pathway, 4 and 5 pathway, are mainly present in non-macrophage cells, in epithelial cells or endocellular cells, because those cells are more frequently encountering bacterial infection to recognize, so that they can recognize LPS. So before I get into the mechanism of pyroptosis, I want to use this movie to show, to illustrate to you what is the real difference between apoptosis and pyroptosis, and in a morphological sense. And you can see in apoptosis, the cells shrink and the cell body fragmented into small vesicles. But the key feature for apoptosis is that the plasma membrane integrity is maintained. So for pure apoptosis, there's no leakage of cytosolic contents, and the plasma membrane integrity is maintained. So, and then not only that, the phosphatidylserine will be flipped out from the inner leaflet of plasma membrane to the outer leaflet. So then that serves as an engulfing signal, eat me signal for macrophages. So apoptotic cells will be engulfed or phagocytosed by macrophages instantly, and which means that apoptosis will not trigger inflammation. It's a clean form, it's a clean death. And on the right side, you see the pyroptosis. Here, we did not add the propylene iodide, so that you can see the morphology more clearly. You see the cells swell, and as the bubbles, big bubbles blowing out from the plasma membrane. Eventually, the membrane undergo lysis, and that releases cytosolic contents, which will trigger strong inflammatory response. So you can see from these two movies, morphologically, apoptosis is shrinking, plasma membrane integrity is maintained, and it's engulfed by macrophages. But pyroptosis morphology is characterized by swelling, membrane lysis, and release of cytosolic contents. And biologically, apoptosis does not trigger inflammation. That's why it's more important and more involved in physiological developmental process, while pyroptosis will trigger strong inflammatory response. So it's more a pathological response, is reduced by bacterial infection or other stress conditions. So what is the execution mechanism? And again, in 2015, Vishwa Dixit's lab and my lab, we independently identified this gene called gastromin D. So we performed a genome-wide CRISPR-Cas9 screen to look for genes that are essential for caspase-1 or caspase-11 induced pyroptosis. So basically, in downstream of caspase-1 or caspase-11, you critically need this gastromin D. If you knock it out, and there's no pyroptosis. And gastromin D encodes a protein about 500 amino acids. The two domains, N-terminal domain, C-terminal domain. These two domains bind to each other. That keeps the protein in the auto-inhibited state in the absence of caspase activation. So once caspase-1 or caspase-11 are activated by upstream inflammatory signals, for example, in response to bacterial infection or other stimulus, and caspase-1, 11 will cleave this gastromin D protein after this FLTD motif within the interdomain linker region. And that will liberate the N-terminal domain. This N-terminal domain will undergo conformational change upon binding to membranes or lipids in the membranes. This along with dramatic conformational change, the N-terminal domain of gastromin D will oligomerize into a larger pores, a very large pores. Then that will be the pores will be inserted into the membranes. You see, there are multiple pores of regular shape and size in this artificial lipid bilayer. And Howard's lab at Harvard determined the cryo-EM structure of the entire gastromin pore. You can see that, and the pore is quite large. And this region, this beta sheet region, will be inserted into the membrane. For each pore, you have about 27 to 28 protomers, which basically is the N-terminal domain of gastromin D that is released by caspase cleavage. Once you have multiple pores formed on the plasma membrane, it will disrupt the osmotic potential. It will disrupt the osmotic potential, and then water will go inside, and the pyroptotic cells will swell and eventually undergo a burst and a membrane lysis. So with the discovery of gastromin D and the mechanistic explanation, we have a complete understanding of what is the molecular mechanism of pyroptosis now. So as I mentioned, that active caspase 1 and 11 will cleave this FLTD motif. So some of you may know that in the caspase biochemistry field, all the caspases are known to cleave after an aspartic acid residue with a specificity to the tetrapeptide sequence in the cleavage site. So that has been a dogma in the caspase biochemistry or structural biology field. So different caspases will recognize different tetrapeptide in their cognate substrates. However, this appears to be not the case for gastromin D cleavage by caspase 1 and 11. In this paper published last year, we found that the caspase, the dimeric caspase 11 or caspase 1, caspase 2, will recognize this C-terminal domain, of gastromin D, and this enzyme substrate recognition is key for active caspase 1 and 11 to specifically recognize gastromin D and then cleave after this aspartic acid residue. And in fact, due to the exocyte-mediated enzyme substrate recognition, the sequence of the tetrapeptide is not important. You can generate a mutation into this FLT motif into triple alanine. You still get efficient cleavage of gastromin D. So this is a feature. Different from all other caspase substrates when cleaved by the caspase. So means that it's a tetrapeptide independent cleavage. So if you are interested in biochemistry, you can look over this paper. Now I'm going to get into the second part of my talk about the role of pyroptosis in antibacterial host immune defense response. So as I mentioned, that pyroptosis was discovered in the context of bacterial infection. If we generate gastromin D knockout mice and infect mice with various types of bacterial pathogens, for example, a chromobacterium violation, this bacteria will be recognized by the NAP NRC4 caspase 1 canonical inflammasome. So the flagellin in the bacteria is recognized by this inflammasome receptor called the NIBS that we identified. So in wild-type mice, this pathway will be activated to induce an immune defense response through gastromin D-dependent pyroptosis. So you see, wild-type mice can survive 1,000 bacterial infection. However, if we knock out any gene in this pathway, the NABs, the NRC4, caspase 1, gastromin D, the mice now are susceptible to the same dose of chromobacterial infection, and the mice will die within 1 week. The knockout mice, like NRC4, gastromin D knockout mice. So if you check out the bacterial load or bacterial replication in the liver or spleen of the infected mice, you see that there are 2 orders of magnitude higher bacterial load in the knockout mice in their liver and the spleen than the wild-type mice. So basically, if you knock out this pathway, you knock out gastromin D, and the bacteria will replicate extensively, and eventually the mice will die out of massive bacterial replication. And the same story, the same is true with another bacteria called Francisella novacidal infection. And this bacteria is recognized by the M2 inflammasome, and the downstream is the same. And the double-stranded DNA will leak out from the bacteria and then recognized by this double-stranded DNA inflammasome receptor called M2. And then you can see that wild-type mice, they can survive the infection. Caspase knockout mice or gastromin D knockout mice, they die out of the infection. They cannot limit or restrict bacterial replication. You can see this in there are a lot of bacterial replicating in the liver of the spleen of the knockout caspase or gastamine D knockout mice. However, for both experiments, we cannot attribute the function or the phenotype solely to gastamine D, the role of pyroptosis, because when we knock out gastamine D, we also block I1 and I18 secretion, because the pore, as I mentioned, the pore actually serve as a conduit for mature I1 beta and IL-18 to get out of the inflammatory cells. There are lots of evidence in the literature that gastamine D not only make pores to kill cells, but also the gastamine D pore is important, is essential for secretion of mature I1 beta and IL-18. So that means if we knock out gastamine D, we also eliminate the cytokine response downstream of canonical inflammasome activation. So to really pinpoint the role of pyroptosis rather than cytokine response in antibacterial immune defense response, we then turn to another bacteria called Shigella fraxinare. And this bacteria is intracellular free living. Intracellular free living, many of the intracellular bacteria, they live or they reside in a vacuum structure. But Shigella fraxinare is very unique. It does not live in a vacuum membrane-bound structure. It's freely live in the cytosol, which means that this bacteria will inevitably leak out their LPS, and then a cytosolic LPS will be directly recognized by caspase 411, and active caspase 411 will cleave gastamine D or trigger pyroptosis. And in this pathway, caspase 4 and 11 do not process interleukin. So we only have cell death response out of this pathway. So this work was just published 3 weeks ago by, and the first author is a postdoc fellow at Zilingli in the lab. And there's really a twist in this story that when we infect mice, either white-type mice or caspase 11 knockout mice, to block this so-called non-canonical inflammasome pathway, the pyroptosis pathway, we did not see a difference, although we expect that LPS from Shigella will be detected or sensed by caspase 411, but we did not see a difference between white-type mice between caspase 11 knockout mice, even for bacterial load. And however, in a control experiment with another similar bacteria called Burkholderia, in this bacteria is also cytosol free living, and white-type mice can survive Burkholderia infection. If we knock out caspase 11, the mice succumb to the same dose of Burkholderia infection. So why is that? It turns out that Shigella, which is different from Burkholderia, and both bacteria will leak out LPS, and that is sort of expected. But for Shigella, it encodes a toxin or type 3 effector protein called asparc-C3. And this protein from bacteria, which is a toxin from the bacteria, it turns out this protein has a very interesting, unique activity of inactivating caspase 4 and 11. If we genetically delete the toxin from bacteria, now the mice can survive the infection. Basically, we delete this toxin, we diminish the inactivation of caspase 11 by the bacteria, and then the bacteria become avalent, or this valence is reduced. Now the mice can survive. The mice can survive out of the bacterial infection. If we knock out caspase 11, now the mice will succumb to the infection. So basically, bacterial replication is the same, supporting in line with this observation. So basically, we can remove asparc-C3 from bacteria. We turn this bacteria into a bacteria that can stimulate a strong caspase 11 gastamine D pyroptosis pathway in the mice. So biochemical studies suggest that this bacterial protein is an enzyme use NAD as a ligand to modify an arginine, a specific arginine in caspase 411. And this modification is brand novel. has never been discovered. So we gave a name as ADP riboxanation. So, and indeed, it took us 3 years to figure out, 3 years of hardcore biochemistry work to figure out this very strange or unique post-translational modification. Anyway, so now we have a bacteria, the OSPC3 deletion bacteria, and this bacteria is, the virulence is reduced, and this bacterial infection will stimulate the mice to upregulate their caspase 11 mediated pyroptosis pathway. So when we measure Shigella IgG in the serum of the infected mice, we can see that in the wild-type mice, when they are infected either by wild-type Shigella or the OSPC3 deletion mutant, with deletion mutant infection, we see a much higher level of anti-Shigella IgG production. And this was not seen in caspase 11 or gastromin D knockout mice. So this genetic data suggests that the caspase 11 gastromin D pyroptosis pathway can promote the mice to mount anti-Shigella antibody response or humoral immunity. So this is the first time to demonstrate that the pyroptosis pathway can activate the IgG production in the adaptive immune system. So this avalent OSPC3 deletion mutant basically serve as a attenuated virulence attenuated bacteria, so give immunization protection effect. If we re-challenge the infected mice with a high dose of wild-type bacteria, you can see that the deletion mutant infected bacteria have a much better protection. So basically, we have like a vaccine-like effect, and this vaccine-like effect is dependent upon gastromin D or caspase 11, because in the knockout mice you don't see that. And for Shigella, there's no vaccine available now. There are two virulence attenuated strains, ICSA and GuarB8 are the two live attenuated vaccine strain currently in development. So if we compare antibody production of the OSPC3 deletion mutant with the two clinical candidate strain, vaccine candidate strain, you see that our mutant actually stimulate a higher level of anti-IgG, anti-Shigella IgG production than the ICSA and the GuarB8 strains, both in black 6 mice and BarbC mice. So if we combine our mutation, OSPC3 mutation, on the background of these two clinical candidate or vaccine candidate strains, you see that the antibody production level is much, much higher now. So this is a double deletion. Then the ICSA and OSPC3 double deletion gives a much, much higher level of antibody IgG production than the ICSA strain. And now we can see a much better immunization or protection effect or vaccine-like protection effect if we combine OSPC3 mutation onto the background of these two vaccine candidate strain. So this experiment not only suggests, for the first time, to suggest that the non-canonical inflammation pathway or the caspase 11 gastromin D pyroptosis pathway have a key role of instruct the mice to induce humoral immunity to promote antibody production. But this data also provide a rationale to design, probably better design anti-Shigella vaccine in the future. So this, as I mentioned, that the knockout, the pyroptosis pathway play a protective role in antibacterial defense. That's why some bacteria have evolved a mechanism to modify caspase 11 and block this pathway. On the other hand, inflammation or immunity is always a double-edged sword. So if the pathway is overactivated or is out of control, it will give a deleterious effect. So if we give mice a really high dose of LPS to induce septic shock or endotoxic shock, in this experiment, we can give mice like a 50 milligram per kilogram. So the LPS will activate caspase 11 gastromin D to induce pyroptosis. The wild type mice, they will die actually because hypoactivation of this pathway. So low level activation of this pathway give a protective effect, but excessive activation of this pathway give a deleterious effect to induce septic shock. So this data suggests that systemic pyroptosis is likely the key for septic shock, which will further induce a cytokine storm response. So now there are efforts in the industry to target the gastromin D to develop antisepsis drug. So as I've talked about gastromin D now, we also have gastromin A, B, C, E. So this is a family of proteins. There are 5 members in human, A, B, C, D, E. And this family of protein all share this 2-domain architecture, N-terminal pore-forming domain, C-terminal inhibitor domain. Normally, the N-terminal pore-forming domain is auto-inhibited by C-terminal domain. But different from gastromin D that I just talked about, other gastroamines are not cleaved by caspase 1 and caspase 11. It could be cleaved by any other protease. So as long as you can cleave within this long linker region, you will activate the protein. So this suggests that pyroptosis, the concept of pyroptosis has evolved. It could have nothing to do with caspase, but the core of pyroptosis, the key feature of pyroptosis is gastromin-mediated programmed necrotic cell death. And this family of gastromin protein is highly conserved in lower organisms, even including coral and hydro. And also, and there recently there are papers showing that bacteria, some bacteria, a group of bacteria even have gastromin-like proteins. And in bacteria, gastromin can also be activated by a protease. And this locus is often next to a CRISPR locus. And here is a pore made by the bacterial gastromin. You see the very beautiful regular shape pores of bacterial gastromin. And the physiological function of this pyroptosis or gastromin pathway in bacteria probably is involved in antiphage defense response. I think there will be a paper coming out soon to illustrate this. And in fungi, there are also gastromin-like proteins. They can also trigger fungal cell death. And in fungi, you have this haploid to fuse into diploid. But if the genetic type, karyotype of the haploid is not compatible, when the 2 haploids fuse together, the diploid will die out of fungal gastromin activation. So in fungi, this bacteria, in fungi, gastromin-mediated cell death indeed protects, serves as a protection mechanism or to discriminate, is the non-cell discrimination against genetically incompatible fungal strains. So now I'm going to talk about the role of gastroamines and pyroptosis in cancer immunotherapy. So as we know that in the apoptosis, canonical apoptosis pathway, if you trigger DNA damage, if you UV irradiate cells or treat cells with DNA damage drugs, which will cause DNA damage, activate the mitochondrial pathway to induce caspase-3 activation and then to have apoptosis. So in HeLa cells, if you give radiation or treat with chemo drugs, DNA damage drugs, you see HeLa cells undergo typical apoptotic cell death. You see the morphology is like a cluster of grapes. However, if we express gastromin E in HeLa cells, this is the second gastromin I talk about. We do the same treatment. You see now the cells did not shrink, but they develop a lot of bubbles and they eventually undergo burst. So basically we ectopically express gastromin E in HeLa cells. Now it is switch caspase-3 mediated apoptosis to gastromin E dependent pyroptosis. And why is that? The reason is that there is a caspase-3 cleavage site in the linker region of gastromin E. In biochemical experiments confirm that caspase-3 can efficiently cleave full-length gastromin E into two fragments, N-terminal pore-forming domain, C-terminal inhibitory domain. Other caspase were not cleaved. If we make a mutation of the aspartic acid residue, so caspase 3 cleave of gastromin E does not involve the C-terminal domain. It only recognizes the tetrapeptide motif. And the aspartic acid is key, is absolutely essential for the cleavage. You mutate the aspartic acid, you block the cleavage. So if we put this D2A mutation back into HeLa cells, you can compare. This is the experiments I just showed you a minute ago. Wild-type gastromin E was switched into pyroptosis. Now this is a D2A mutation. So on the left, you will see the cells undergo pyroptosis, right? However, with the D2A mutation, the cell death goes back to apoptosis. So this suggests that it is the cleavage of gastromin E by caspase 3 that converts or switch apoptosis to pyroptosis. In fact, pyroptosis is much faster than apoptosis. It only requires 1 or 2 hours, but apoptosis will take more than 4 or 5 hours. So, everybody knows that caspase 3 induces apoptosis, but now we are showing that caspase 3 can also induce pyroptosis. So why is that? Why it is inconsistent with lots of literatures? So it turns out that cancer cells or cell lines, we do all the experiments in cancer cell lines. In cell lines, for example, we have profiled about 100 different cancer cell lines. Very few of them express gastromin E. Most of them do not express or express very low level of gastromin E, just like HeLa cells. So, it turns out that during tumorigenesis, there is a programming, there is an epigenetic reprogramming that drives epigenetic silencing of gastromin E during tumorigenesis. However, we have normal cells in our body those are not cancer cells. Here I'm showing you 5 normal human cells. You see 3 of them, like epidermal keratinocytes, or placental epithelial cells, or umbilical artery smooth muscle cells. You see they express endogenously gastromin E. If you treat those cells with doxorubicin or give them UV or cisplatin, and those cells will all develop a pyroptosis. You see those big bubbles, characteristic big bubbles, right? For the negative cells, gastromin E expression negative cells, you will go into apoptosis. So this data suggests that DNA damage chemotherapy or even radiation therapy used in cancer treatment in clinics, and those approach induce the cell death not only involves apoptosis, which is the traditional view, but may also induce pyroptosis in the real world. So just use this cartoon to summarize that cancer cells, most of the cancer cells have silenced the gastromin E expression. So you have this chemo drug treatment induced caspase 3 activation, and those cells die out of apoptosis. However, the normal cells, they have some of them, not all of them, some of them, they will have gastromin E expression, they will die out of pyroptosis. So what's the function of this pathway, gastromin E mediated pyroptosis? So it will trigger inflammatory damage. It will for those chemo drugs, and it will also stimulate immune response because, as I mentioned, that pyroptosis is the most pro-inflammatory form of cell death, and I'll go back to this point later on. So when we profile gastromin expression in those 100 cancer cell lines, we found that 3 of them, not only gastromin E, gastromin A and gastromin C are also silenced in cancers. Why is that? In this paper published last year, is a collaboration with the chemical biologist, Dr. Zhibo Liu at Peking University. So Dr. Liu, basically, he designed and developed a chemical biology, by orthogonal approach that allow us to deliver gastromin selectively into tumor cells, cancer cells in vivo. I mean, in vivo in the mice. So basically, he has a chemical linker this chemical linker will be conjugated to gasdermin and then to nanoparticle, and the cancer cells will uptake nanoparticles, and he has a small molecule to cleave this linker. This small molecule is also enriched, has a preference to target cancer cells, and then cleave this linker, release gasdermin to make pores, to induce a pyroptosis, and then further kill cancer cells. Basically, he gives us an approach for tumor selective protein delivery and release from nanoparticle conjugates. We use that to induce gasdermin activation. So in this 41 breast cancer tumor model, at day 6, 9, and 12, we give the mice we give the mice the nanoparticle conjugated gasdermin and then induce gasdermin activation for 3 rounds, right? At day 6, 9, and 12. And then we see that there's almost a complete remission, a regression of the tumors. We did a lot of other controls. I'm not going to talk about that. Statistically, you see there's a dramatic tumor regression response after 3 rounds of pyroptosis induction. And we also see this, observe this in other tumor models, syngeneic tumor models. So you might think that, well, you kill cancer cells, right? You don't see tumor growth anymore. So that is true, but it's not completely true. And the reason for that is induction of cancer cell pyroptosis by this chemical biology approach is not that efficient. We only can induce about 30% of cell death or pyroptosis in the cell, even in the cell culture system. And in vivo, in the tumor-bearing mice, we can only kill about 10% of cancer cells. So then what's the rest of the 90% of tumor cells? So we then do the experiments in the nude mice, in the asystemic T-cell deficient mice. You see there's no tumor regression response anymore, right? In Y-type RBC mice, there is a nearly complete tumor regression, but in the T-cell deficient mice, there is no tumor regression. So this data clearly suggests that a few pyroptosis would trigger inflammatory response in the tumor microenvironment, and then that will remodulate the microenvironment and eventually activate the T-cell induced anti-tumor immunity. And this reminds us that another phenomenon, for cancer immunotherapy or cancer immunity, we now trace the history back to this doctor called Bill Cowley in 1983. And he found that some of the severe cancer patients can be self-cured from bacterial infection. He later on did some sort of like clinical trial experiments using so-called Cowley toxin. So streptococcus is a bacteria, right? A bacterial infection, in that case, it seems that bacterial infection triggers an inflammatory response or immune response that eventually allow the cancer patients to cure the cancer autonomously or on their own. You might remember at the beginning of my talk that pyroptosis was also originally discovered in bacterial infected cells. I don't know there's a connection or not. There's just accidental happening here. There might be a connection, so we will see. And I think I'll skip the detailed immunological analysis. Okay, and in the previous experiments, we did 3 rounds of induction of pyroptosis. If we reduce the 3 rounds of pyroptosis induction or gasdermin delivery into 1 round, we did not see the tumor regression response in this 41 syngeneic model. And this 41 tumor model is known to be a cold tumor, right? It does not respond to the checkpoint blockade and the PD-1 therapy. However, after even one round of pyroptosis induction, we treat the mice with anti-PD-1 antibody. Now there's a dramatic tumor regression response. So this data suggests that a little bit of pyroptosis will inflame the tumor, convert the cold tumor into hot tumor that now can be responsive to checkpoint blockade therapy. So together, this data also suggests that why we now in clinical trials, we see efficacy of a combination of therapy between checkpoint blockade and chemotherapy or even radiation therapy, right? So a lot of companies are looking for combination therapy. And the best success now is combination with chemotherapy or radiation therapy. As I mentioned, that chemo and radiation will trigger pyroptosis based on our work on gastromin E. And this, the activity of antitumor immunity triggered by pyroptotic death also explains why during tumorigenesis and cancer cells tend to silence gastromin expression if they can, if it could afford. Because if they don't silence gastromin expression, if gastromin gets activated, even accidentally, it may trigger a potent anti-tumor immunity response. So in the last few minutes, I'll talk about another work we published last year. So in this work, we show that even, in fact, gastromin-mediated pyroptosis also plays a role in normal anti-tumor immunity. So why we ask this question? So for this group of audience, we know that cancer immunity eventually converges on activation of CD8 positive T cells, or the killer cells. The killer cells will deliver granzymes into target cancer cells. The activity of those granzymes will eventually induce cancer cell death. The 5 granzymes in human, right, 11 in mice. And those granzymes are not only responsible for killer T cells, but also for NK cells, natural killer cells, to kill target cells. And the textbook will tell you that, the immunology textbook will tell you that granzymes will kill target cells by apoptosis. Is this always true? So in these experiments, we use NK cells to kill 293 T cells. So this is a cell killing assay. Or we load a fluorescence dye, green fluorescence dye into 293 T cells, and this dye cannot get out of the 293 T cells unless the cells are lysed, unless the cells undergo lytic death. So here you see the cell 293 T cells looks green, and there's a small NK cells. NK cells will then attack 293 T cells, but you did not see leakage of green fluorescence, which means that there is no lytic cell death. 293 T cells do not express any of the 5 gastamins. So no gastamin in 293 T cells endogenously. So if we express each of the 5 gastamin into 293 T cells, we found that in the presence of gastamin B, now when NK cells attack 293 T cells, the green fluorescence will leak out. And at the same time, propylene iodide was uptaken into the dying cells. So this data suggests that expression of gastamin B in 293 T cells convert the killing of 293 T cells by NK cells from apoptosis into pyroptosis. So the underlying reason is very simple, that granzyme A, among the 5 granzymes, 5 human granzymes, specifically granzyme A will cleave gastamin B into 2 fragments, N-terminal pore-forming domain, C-terminal inhibitory domain. And the cleavage is also in the long linker region, and then release N-terminal pore-forming domain and make pores and induce pyroptosis. So if we, here I'll show you 3 cell lines, they endogenously express gastamin B, not like this artificial 293 T cells. And those are all from the gastrointestinal system, cancer cell lines. You see the high level of gastamin B expression endogenously. If you treat the cells or give the cells granzyme A, you see big bubbles, pyroptotic bubbles coming out. You see cleavage, very efficient cleavage of gastamine B, endogenous gastamine B by granzyme A. And the CAR T cells it basically is a hypoactivated killer cells, taken from patients, and we add the CD19, sort of like a warhead, right? And then you see if the target cells express gastamine B, the CD19 CAR T cells will also induce pyroptosis. You see this pyroptotic bubbles, right? You see the leakage of the fluorescence and cleavage, efficient cleavage of gastamine B. And if we make a mutation, the lysine mutation to block the cleavage, you block the pyroptosis, right? And the TCR-T cells is the same story. I'll just skip this. So basically, if the target cells express gastamine B, TCR-T cells, when they can recognize the targets, the melanoma cancer cells, it will also induce gastamine B cleavage and then induce pyroptosis. Or just to summarize that, the CD8 positive killer T cells will produce interferon gamma. We also found that interferon gamma can potently stimulate gastamine B transcription. You see that many of the cancer cells, normally you don't detect gastamine B expression, but if you treat them with interferon gamma or even sometimes TNF alpha, you see a dramatic transcriptional induction of gastamine B. So interferon gamma acting on its receptor and then will induce transcription of gastamine B. And the gastamine B gets cleaved by granzyme A delivered from the killer T cells, CD8 positive T cells. And the granzyme A cleave the linker region of gastamine B and to release the N-terminal pore-forming domain and then make pores, and the cancer tumor cells die out of pyroptosis. The pyroptotic death will further amplify the immune response, will further induce antitumor immunity, if you recall the nanoparticle experiments I just showed you. So this, as a result of that, we also see this pathway of interferon gamma, gastamine B, granzyme A, has a synergistic effect with the checkpoint blockade therapy in this CD26 colon cancer model, in syngeneic model in mice. So basically, if you have gastamine B, you convert apoptosis into pyroptosis, and that has a synergistic immune antitumor immune response with checkpoint blockade therapy. So this take together suggesting that gastamine, like B or even E, may serve as a biomarker for cancer immunotherapy. If we can develop small molecules to activate a gastamine independent of anything else, and that might be a potential therapeutic approach. So I just acknowledge the student and postdoc in the lab who did all those work. Jianjin originally identified gastamine B by CRISPR screen, and Jingjing, a former postdoc fellow, did the pore-forming work and the structure work. And the Yupeng, Wenqing, Xu Yan, they did a gastamine E cleavage by caspase-3 work. And Sun Qi and Kun Wang did the structural work on caspase, the tetrapeptide independent recognition of gastamine D by those caspase. And the last piece of story was done by Zhiwei Zhou and Huabing He in the lab. And the nanoparticle work was collaboration with Qingyang Wang, the graduate student of Dr. Zhibo Liu's lab. Feng Shao made a lot of knockout mice for us. I'll stop here. Thank you.
Thank you so much. This is really fascinating. I think there's a lot of data, but I don't feel heavy at all. I mean, it's really interesting. So let's start the Q&A session. If anyone have question, please put in the chat box or just raise your hand. I can put your question to Feng. So I guess I'm going to start to kick off. Two parts, actually. I'm not really expert in innate immunology, but I just have a very basic questions regarding one is the data you show with the HeLa cells that caspase-3 can cleave the
Mm-hmm.
gasmase E.
Mm-hmm.
But I think there's this how to say a two-direction balance in terms of apoptosis and pyroptosis.
Mm-hmm.
So how the cell or how the signal determine which is dominant for the cell death? Because I mean, some of the cells probably possess both of the signaling pathway.
Yes, that's a good question. So basically, when you have caspase-3 activation, you could go towards apoptosis traditionally, or you could go towards pyroptosis. I think it essentially is the expression level of gastromin E in the cells, in not only HeLa cells, in any other cells. If you don't have gastromin E expression or extremely low, and the cells will just go into apoptosis. But if you have high level of gastromin E expression, for example, like primary cells in like a primary keratinocytes, they have very high level of gastromin E expression. They will just go straight into pyroptosis, because pyroptosis has a faster kinetics, right? And there is a, in real world, there is probably is more like an intermediate
Right.
mixed situation. So you don't have enough gastromin E expression, and you see apoptotic response at the beginning. But then with the time going, apoptosis take 5 or even longer time. With time going, you just accumulate the N-terminal cleavage fragment of gastromin E. And then at the end of apoptosis, you see bubbles coming out and the cells undergo burst. And previously, this has been observed previously quite a lot. And people call it secondary necrosis after apoptosis. So I think that's probably, in the real world, it sometimes is a mixture. Yeah.
But
It is determined by expression level of gastromin E mainly.
But like you said, the gastromin E can accumulate during the process of the cell death.
Yes.
But
Mm-hmm.
is that possible during this process? Somehow there's extrinsic signal can transcriptionally also enhance the gastromin E?
That's a good question. We haven't found there could be a transcription signal. So in the literature, there could be transcriptional induction or epigenetic. So because cancer cells, if you take cancer cell lines, it's very hard to see expression of gastromin E because the promoter of this gene undergoes a massive methylation to silence the expression of gastromin E. So I think that makes sense. The tumor cells, they want to escape. They want to escape those kind of killing. It's
Sure.
basically it's an inflammatory killing, right? Yeah.
Yeah.
There could be a transcription in response that has not been discovered yet. Yeah.
Okay. I think I'm going to go ahead to call the name. If you want to ask question by yourself, are you welcome? Liu Bingxu, you want to go ahead to ask question yourself?
Oh yeah, that would be great. Thank you for the great talk. So basically, and there's a lot of amazing these cancer models you have showed, and for the cancer model you have showed, when you it's amazing when you just switch them to apoptosis to pyroptosis just by just poking a hole on the cells, and it just gives a much better response. And what do you think is actually, inducing a better like immune response? Is it just you release more of intracellular new antigen so the DC cells can do a better job to stimulate T cells, or or maybe there are other tonic intracellular cytokines that actually got released during this process? It'd be good to know your thought.
Yeah. Good question. So there is a concept called immunogenic cell death, right? And pyroptosis clearly is the most immunogenic form of cell death. So it could, it certainly, I think there are data showing that it could promote the release of neoantigen and for presentation to dendritic cells. If you recall, I overload you guys, it's just too much stuff. If you recall the bacterial infection work, I showed that beginning that the pyroptosis pathway clearly can promote antigen presentation, even in the context of bacterial infection. So that could be one reason. I think there are definitely other immunological mechanisms. So the dying cells will release some cytokines or some dams that will remodulate the immune microenvironment. So we're working on that, but it's very difficult. You know, it's very difficult. Yeah.
But do you think there's
right.
this?
Yeah, go ahead.
Sorry, you think here is not L1 or L18 because supposedly, for example, in the nanoparticle experiment, you just did right.
Very good point. I don't think it's I1 or I18. So historically, pyroptosis was discovered in this canonical inflammasome pathway, caspase-1 activation, and caspase-1 activation will trigger will process interleukin-1, interleukin-18. So a lot of people read some older literatures all think that pyroptosis is just associated with I1 or I18, and that has been I think that concept has been changed because with the discovery of gasdermin, you know, as long as you have gasdermin activation, right, you may involve caspase-1, you may not involve caspase-1. So yeah, I think in the case of the tumor data I showed, I don't think it's the I1 or I18. It's probably some other cytokines and some dams. You know, pyroptosis will release ATP. You know, ATP clearly has a very important role in modulating the microenvironment. Yeah.
Yeah, and if I may, for the privilege, that following question. So when you are saying there's basically the level of gastronomy E is really important to determine if it's actually, you know, doing apoptosis or pyroptosis. But do you think here is more of a limitation of a gastronomy E level, or you think it's actually, you know, something related to, for example, the enzyme activity? For example, coenzyme A is actually a good enzyme to cleave the gastronomy E. Like for example, if you have more efficient enzymes.
I think, yeah, I'm glad to hear these questions, which means that my presentation is successful. So yeah,
it's really great.
Right.
Thank you.
So, it's not only determined by the level of gasdermin level, it's also determined by the granzyme activity. You're absolutely right. So eventually what matters is how, you know, the level of the N-terminal cleavage fragment you can accumulate in the cells within a period of time, right? That is determined by the level of substrate and is also determined by the activity of the enzyme. You're absolutely right. Yes.
Yeah, you are thinking just the final deterministic factor is active gastronomy concentration. Which, yeah, okay. Thank you for this great talk. I really appreciate it.
Okay, so next one is Gefei. You want to go ahead? I saw you just turn on your camera.
Yeah. Thanks, Professor Shao, for the great talk. So I wonder if have you done any work for gastronomy E in the human tumor samples or PDX models? If not, are you planning to do anything?
We have not done. Yeah. So if you look at the TCGA data, we actually profile a little bit on primary tumors. So clearly, there is the silencing effect of gasdermin E even primary tumors, not only just cell lines. And besides that, we haven't done anything on that. So yeah, you're welcome to do that.
Yeah, I'm actually from pharmacology, from industry, so it'll be very interesting to have some collaboration and our research in this field. Thank you.
Sure. Yeah, sure. I'll be glad to help you guys. You know, if you need any information or whatever. Yeah.
Thank you.
Okay, so next question from Jay. I think Jay's already logged out, but I still gonna recite here the question. What is the relationship between the MLKL mediated necrotosis and then GSDM mediated pyroptosis?
Yes, MLKL is the most downstream component of the necroptotic pathway. So the necroptotic pathway is activated when you block caspase activation, and at the same time, you stimulate the TNF receptor response. So, and then that will, basically from the receptor, you activate caspase 8. At the same time, you block caspase 8, and that somehow drives a complex formation between RIP1, RIP kinase 1, and RIP kinase 3. And then RIP kinase 3 will phosphorylate MLKL. The phosphorylated MLKL will move to the membrane, and then the cells die out of necroptosis. So there's not much overlap or crosstalk between pyroptosis here.
Okay. So next question is from Highway. You want to go ahead to ask question? Okay, I'm gonna just read the question. Why pyroptosis can go faster than apoptosis?
Well, I think intuitively it should be faster, right? Because
Yeah.
the downstream is only one step. You cleave it, it goes to plasma membrane, make pores. But for apoptosis, you have caspase activation. So basically, downstream of, I think there is a misconception in the textbook or in the literatures from the 90s that we deem caspase activation, like particular caspase 3 activation, as the executional step in apoptosis, which is not correct. Which is not correct.
Sorry, go ahead.
Downstream of caspase 3 activation, there are multiple events. For example, you have DNA fragmentation, right? And caspase 3 needs to cleave an inhibitor of endonuclease. You have PS flipping, right? And the caspase 3 needs to activate some scramblase or flippase on the plasma membrane. Then you have PS flipping, and you have the cells shrink, right? So the cytoskeleton is disrupted downstream of caspase 3 activation. And probably some cytoskeleton protein is cleaved by, inactivated by caspase 3. So there are multiple events. There are multiple events downstream of caspase 3 activation. And in fact, if you really think deeply about apoptosis, which event is the point that you can say is a determined point for cell death? I think it's more like a philosophical question,
Yeah.
right?
Right. Okay. So so next question is from Joe. You want to go ahead, ask question? Okay. I'm going to read the questions. Okay. So go ahead, Joe Gang. Yeah. I thought it was
hi, very elegant work. That's very impressive. Actually, you already touched upon the question I want to ask. The first question is that whether, what's the mechanism pyroptosis induce stronger anti-tumor immunity? You mentioned probably it's because, immunogenic cell death. So, and also you showed that CAR T cells in your CAR T cell model, your CAR T cells or T cell transgenic T cells can also induce pyroptosis in tumor cells. In that case, so is there a mechanism that directly affect the CAR T cells or the donor T cells? Or it still has to go through the endogenous immune system. So that's one question. So related to that question is that, for immune checkpoint block therapy, I think it's very effective in a small fraction of patients. So in those patients, those responders, do you think that, somehow the immune cells induces pyroptosis in the tumor cells? That's why, it's so effective. While the non-responders, maybe, it's just a regular, apoptosis in the tumor cells. So does that mean that, if you stratify the patients to say, if some patients, whether their tumor cells, somehow their biopsy shows that the tumor cells express gastromin B, and then those patients will be more likely to respond. So that's a second. But third question is that, you mentioned interferon gamma can promote pyroptosis. Also, you showed side-by-side interferon gamma and TNF-alpha, but you didn't go through TNF-alpha. So does TNF-alpha promote pyroptosis, or it inhibits pyroptosis? That's my questions.
For your first question, basically, is still the immunological response downstream of the pyroptotic death, right? So yeah, I mean, we don't know the complete mechanism out of that. The dying cells will release ATP, will also release HMGB1, and there are probably other cytosolic dams leaking out from the dying cells. And for the CAR T cells, and for the CAR T cells killing, and indeed, the pyroptosis actually, that killing is not good for therapy. And the reason for that, because now for CAR T, we only have this CD19 or BCMA CAR T, which was used to treat the lymphoma, right? Which is not solid tumor. So for solid tumor, and the pyroptosis probably is give a better or beneficial immunological effect. But for killing of the blood, the lymphoma, it's not a solid tumor, it's a circulation system. And that kind of killing, when you have pyroptosis, you leak out lots of dams, and probably may induce a cytokine storm, because for this particular target cells, I mean, death is death, right? Whatever, pyroptosis, apoptosis, it's the same. The difference is the downstream. And if the cancer cells is not in this microenvironment, right, like lymphoma, in the circulation system, and those leakage of dams or other pro-inflammatory factors might not be good. So that's for your to answer your first question. The second question, we certainly think that those gastromin could be biomarkers for immunotherapy. So, but I want to emphasize that immunotherapy or cancer immunity is a very complicated process. I mean, this is just one step, right? We also have a lot of other steps. Even if you have gastromin there, you may have lots of Tregs or whatever. You still can block it, right? So, I mean, we are only showing the concept, proof of concept in principle, but when it gets to real world, to really treat patients, you have to, you can stratify the patients as you mentioned, but whether it will be like that simple scenario that all the positive ones will respond, all the negative ones will not respond. I guess you just have to do the experiments. You just have to do the trials, right?
Yeah, how about TNF-alpha?
I'm not sure if the
applications. Yes, so most we profile about 40 or 50 cell lines. We don't see that wide expression of gastromin B. And then the reviewer just challenge us that this is probably not that important. All the pathway you show is correct, but it's not that important because you see among the 50 cell lines, there are only a few of them have gastromin B expression. Then we sort of, well, there could be some regulation. We just we collected a panel of cytokines, not only interferon gamma, TNF-alpha. We tried a lot of other cytokines, and there's a dramatic transcriptional induction. So I mean, this is the transcriptional induction. The transcriptional induction is not sufficient to induce cell death. you need a second signal from granzyme A. If you don't have granzyme A, you just express the protein, they will not die. The protein is activated by cleavage. But transcription of gastromin B is controlled or is induced by interferon gamma on TNF alpha. So TNF alpha, for example, if like K562 cells, interferon gamma still can induce transcriptional induction, but TNF alpha cannot. So we don't know the reason for that. Some cells can respond to both transcriptional induction of gastromin B. Some cells only respond to interferon gamma, and the extent also varies. Like this cell line, even interferon gamma is not that efficient, but for this is HT29, interferon gamma is dramatically efficient. So for the signaling pathway of this transcriptional induction, we haven't gone much on that direction. Yeah.
Great, thank you. Appreciate it.
Okay, so I think before I go further, I want to check with Feng. You're going to be bombed with the questions. I just want to be sure you have enough time because I don't want to delay your any other events.
Oh.
Yeah, I mean, there's a lot of people want to ask questions. You see, after 25 minutes, we still have 230 people online, which is quite a record. So I want to make sure that we don't delay any your business.
I can go 10:30. I have another thing coming out. Yeah, 10:30. We'll have
another 10 minutes. Okay.
Yeah.
So everyone, we have another 10 minutes. I'm going to try to squeeze some questions. If you are not named, please just I think you can send email to Dr. Shao. He'd be happy to answer the question. Okay, so next question is from WK. So the question is, in the study you found that gasmin B was involved in cancer lysis. So the question is whether gasmin D, gasmin E, and the other members are also involved in this process. Since there are many gasmin molecules, what is the difference between these different molecules in those induced pyroptosis?
Yes, good question. We haven't found in this immune cell killing, like T cell, NK cell killing context, we haven't found any involvement of gastromin D. But we cannot exclude the possibility. So, but for gastromin E, there is a paper, sort of like a back-to-back paper from Judy Liebman's lab at Harvard, and they showed that gastromin E, we found that gastromin E is cleaved by caspase 3, right? And among the 5 granzymes, actually the best studied granzyme is not granzyme A, is granzyme B, another granzyme. So granzyme B is known to activate caspase 3. So they have a paper connecting granzyme B, caspase 3, and gastromin E, and also in the tumor context. So I think that granzyme B is probably the most dominant granzymes. So that somehow explains why gastromin E is much more frequently silenced in cancers than other gastromin, because gastromin E can be activated caspase 3, right? Caspase 3 can be activated by granzyme B or even many other signals. Yeah, so that's about gastromin E. Gastromin A and C, there are very little studies on that. We don't know much about that yet.
Okay, so next one is C06. Do you want to ask question yourself? All right, so I think this is going to be the last question. Everybody else, I'm sorry. So Feng is really popular. You have to find another time to send him an email about your question. So I think C06 asked question, pyroptosis could turn cold tumor to hot tumor. How about pyroptosis in inflammation associated cancer like colon cancer?
So the experiments I showed, basically, we have, we already have a tumor, right? And then if you, in some way, you can enforce the tumor cells to undergo pyroptosis or whatever, you trigger an immune response. I think we have to distinguish it, or have to distinguish the therapy, which happens after you have a big tumor growing there,
Right.
and the process of tumor genesis, from a single cancer stem cells growing into a big tumor. In that process, there could be a possibility that the inflammation, a little bit of inflammation triggered by pyroptosis may have a promoting effect. Inflammation is complicated, right?
Yeah.
There is a theory that cancer-associated inflammation, sometimes you need a little bit of some kind of inflammation to promote tumor genesis. I think that is possible, but you just have to study it in a particular context. It's hard to have a generalized concept for everything.
I agree. Okay, so I'm gonna let you go. So there's still a ton of questions.
Thank you, Fran.
Yeah, thank you so much for doing this. I really appreciate it. So hopefully I'll see you soon in person. Thank you very much.
Thank you.
Yeah, thank you.
Bye.
Thank you.
Yeah, thank you.